last talk in the session, and the laser doesn't. I am Rochish, as I introduced myself in the morning, and I'm in the Department of Chemical Engineering for last 20 years now at IIT Bombay. So I'll talk about electroporation. It's a little at odds with the theme of the conference, but I think there will be something interesting for you to appreciate in this. So electroporation. So this is the brief outline of my talk, which is essentially what is electroporation, and I'll tell you why we are interested in giant vesicles as a system to understand biophysics of electroporation, and I'll try to explain this with some sprinkling of results from my group using giant vesicles and electroporation. So I gave part of my talk as the first in the morning, so that I can save on time now. So I already introduced this to you, and so essentially what you have is bilayer membranes, as we know, are the objects of interest here. And what I would emphasize for my part of talk is that the bilayer is made up of hydrophobic tails at the center of the bilayer, with the hydrophilic heads on the other two sides. And I'm going to apply electric fields. So from an electrical angle, the bilayers, the hydrophobic tails, are essentially insulators or dielectrics. So they are not going to allow polar ions to go through them. Okay, so that's something we have to keep in mind. So what essentially happens is, if you have a cell and you want to put in hydrophilic drugs inside, or if you want to put any polar molecule inside the cell, it's difficult because of this barrier. Okay, and so there was this nice technique that came up called electroporation, in which you punch micropores, nanopores in the cell membrane by applying very strong DC pulses. Okay, so you're talking about DC pulses of the order of around 0.5 to anywhere between 2 kilovolts per centimeter and even higher. And you apply them in terms of pulses, which are of duration of around 100 microseconds to 1 millisecond to 2 milliseconds. So you put the pulse on and then you put it off. And when you put it on, when you put the pulse, there are these tiny pores that are formed in the membrane. And when the pores are formed, because of electrophoresis, you can have drug molecules enter inside the cell. Okay, so that really is the process of electroporation. And that's what we want to understand. Okay. So I'll just give you a very, very simple mathematical analysis first to get you started. So electroporation involves essentially applying electric field, as I said. So you can look upon this as an RC circuit. I just cannot control this cursor for some reason. It's really difficult. So you can look upon this as an RC circuit, and if you connect an RC circuit to a voltage source, the capacitor gets charged. The two plates of the capacitor gets charged, and they get charged over a time scale over the RC time scale. Okay, so the resistance into the capacitance gives you a time scale, and the voltage builds across this capacitor in this manner through a first order process. Now, if you have a biological cell under electric field, it's essentially the same system, where the resistance part is played, the role of the resistance is played by the resistance in the extracellular and the intracellular liquid, the aqueous liquid. And the capacitance is essentially that of the cell, because the cell is made up of hydrophobic tails, right? So the bilayer. So you have the cell capacitance, which resists formation of potential across it. And so similar to this RC circuit, the potential builds on this cell through this equation called as the Schwan equation. This is a celebrated equation. And it tells you that when you apply an electric field E0, you get a transmembrane potential Vm across the cell. And since the cell or the vesicle is spherical, you see that maximum transmembrane potential is obtained at the poles, and it will be 0 at the equator, and it has a cos theta kind of variation. And this potential builds over what is called as the membrane charging time, which is given here for a biological cell. And sigmas are the conductivities, intracellular, extracellular conductivities. C is the cell capacitance, which is roughly of the order of 100 microfarads per centimeter squared. Okay, so that's the essentials. Now, if you model this, you really see this. So, for example, if I plot the transmembrane potential versus time, At the poles, you see that a first order increase happens and it saturates. And at 51 degrees Celsius, for example, if you plot it somewhere here, the transmembrane potential is lower. Okay, so you follow the dotted lines. Okay, so this is how the transmembrane potential builds. So now what happens next? Now, once the cell develops the transmembrane potential, then if you consider this part and zoom it out, you essentially have a bilayer, and under the action of electric field, the ions have accumulated across this bilayer. Okay. Now, because of these accumulation of ions across the bilayer, which leads to development of transmembrane potential, electric stress builds in it, which is given by this. It's called as Maxwell stress. And so then electric stress leads to deformation of this membrane and development of a elastic stress in this membrane. Okay, so this is the expression for the elastic stress. And as Samir had mentioned in the morning, if it reaches a value of lysis tension, which is roughly around 5 millinewtons per meter, the cell can give way to pores. So if you just balance these terms, it's a very rough calculation. You get that at a voltage of around 1.6 volts, your pore should form. And if you do very deep theoretical analysis, and in fact the convention in the literature, now everybody accepts that a transmembrane potential of around 1 volt is can lead to pore formation. Okay. So we redo the equation, we resolve the model now including poration. And what you see is the transmembrane potential builds according to Schwan equation. But as soon as the transmembrane potential value reaches a value of around 1 volt, poration starts. Once the membrane porates, it's no longer a dielectric, it becomes a conductor. And so it's like shorting of a membrane, shorting of a dielectric, so the transmembrane potential falls down. And so the poration is controlled. Okay. That's really the physics of the problem. Okay. So now, what are the energetics of pore formation? I thought I'll just quickly go through this. So if a pore is formed in a bilayer membrane, so this is a bilayer membrane, and you form a pore, then there is an energy cost to it. Okay. So these are hydrophobic pores. Then the lipid molecules turn around, they form hydrophilic pores. Okay. And you are talking, so I'll run you through this. So what are the energetics of a hydrophilic pore? There is an edge tension. The edge tension, which I think Mahesh also talked about in the morning, is essentially the, I cannot hold this. I think I'll just go. Can I have this other mic? I think I'll just go. So what you have are your lipid molecules in contact with water, which leads to the edge tension. Okay, and so it doesn't want to have the edge tension. So you have this energy cost, which is the edge tension energy. Now, if you stretch the membrane, it's easy to form pores, and so there is a negative term associated with tension. If the membrane is tense, you can form a pore more easily. Then you have the third term, which is the positive energy. So if the pore becomes very small, these lipids have frustration turning around, and so the energy cost goes up. Okay. So you see here that the energy of the pore goes up as the pore size decreases too much. And then if you apply electric field, there is this negative term, which means your energy of the pore goes down if you apply electric field. And that's because you are replacing the hydrophobic tails, which have a dielectric constant of 2, with water, which has a dielectric constant of 80. So these are really the energetics of the pore. And if you plot it, you see that there is a minima, which turns out to be around 0.8 nanometers. So essentially, you will have abundance of pores of around 0.8 nanometers. If the pore tries to grow in the absence of electric field, you see a huge energy barrier of the order of around 300 kTs. And therefore, normally cells without electric field will have huge resistance to formation of pores. But as soon as you apply electric field, I have shown calculations roughly for 0.6 volts of transmembrane potential. You see that the barrier reduces significantly to a value of around 12 kT or so. And so now the pores can easily grow. The pores can grow really fast in this regime. And so you would want to switch off the field. And as soon as you switch off the field, it may go here and come back. Okay. So you can have controlled formation of pores just as defined by this equation by suitably adjusting two things, the strength of the electric field and the duration of the pores. Okay. So that really is the heart of electroporation as a technology. Now, I'll give you three examples from electroporation, which is in biomedical use. The first is what is called as electrochemotherapy or ECT. Now, this essentially is administering two types of drugs, cisplatin and bleomycin, for skin cancer. And this is what it traditionally evolved as. And it involves formation of very tiny pores of the order of around 1 nanometer to maybe 10 to 20 nanometers, applying very short, medium-sized electric field. So you look at 100 microsecond of pulse times, and you deliver it to the tumor site. And then you see that there is huge uptake, there is very nice uptake of the anti-cancer drugs cisplatin and bleomycin in conjunction with electric field. So that really allows you to reduce the dosage in anti-cancer treatment. Okay. So this was taken up in Europe as an ESOP project in 1991, and by around 2004 to 6 they had guidelines for electrochemotherapy (ECT). Okay, and so this is an example from their conclusion paper, where if you have a lesion, skin cancer, so it's melanoma mostly, and so most of the development in this area happened over skin cancer, skin melanoma, because the Westerners are affected by skin cancer. We in the East may not be. And the reason for development was as simple as access to the skin, to the lesion. Okay, so the lesion is on the skin, you just put the electrodes here and you can do the therapy. And that's how it all developed. So what you do here is give an anesthesia, inject cisplatin, bleomycin. Then you have this electroporator here, and you see the electrodes being put in here, and you apply voltage, you apply short pulses, and then after therapy you see something like this. And it has been remarkably successful in the West in curing skin cancer. Okay. And so finally the protocol that is now used by everyone there is one session of electrochemotherapy will be 8 pulses. So you put 8 electric pulses, each of 1 millisecond duration, of strength 1 kilovolts per centimeter. And then your one session is over, and you may have a couple of such sessions, and then your treatment is done. Okay. So the other example is electrogene therapy. Okay. So in electrogene therapy, as Dr. Arvind mentioned, your idea is to put a gene into the cell with a DNA mini plasmid as a vector here in this case. And the electric field parameters now are completely different. The mechanism also is quite different. Okay. So you are really targeting the movement of the DNA plasmid, which will be of the order of 40 to 50 nanometers, to the surface of the cell using very low electric fields. And then the plasmid electrophoretically migrates to the cell and gets attached to the cell. And then over a period of time, through these processes of endocytosis and so on, the DNA gets into the cell, and then into the cytoplasm, then to the nucleus, and then you have gene transfection. Okay. And I have picked up one example of a mouse muscle cell, and here is the control in which you don't do electroporation, and here you do electroporation, and you see muscles formed because of the gene therapy in this case, and the dye which is associated with the gene is also expressed everywhere here, right? So that's the electrogene therapy. Here you are using low electric fields for long duration of time. The third is irreversible electroporation, wherein you actually do ablation of cancerous tumors, and but it's a non-thermal cancer. It's coined as a non-thermal tumor ablation process. And here what you do is put really long pulses of the order of 2 milliseconds or so, and fairly strong electric fields of 1.5 kilovolts or so. And here what you really do is completely ablate the cell, but in a non-thermal manner. Okay. And the cells would die because of annihilation. Okay. So I have one example here again from the literature where you see irreversible electroporation done for treatment of cancer tumor. And my ex-PhD student Sudeep is working in TCS now, and he develops FEM-based software to guide surgeons to tell them what kind of electric fields you should use for for irreversible electroporation. So it's actually being practiced in medicine, in the medical fraternity, mostly in the West. So, so I'll just give a glimpse of a few of the research that we do here in my lab. So I'll put the pointer off because I want to play some videos. Yeah. So this work is by the session chair, Rakesh, and he did PhD with myself and Samir some years ago. And he developed a method called dissipative particle dynamics to simulate biomembranes and electroporation. So he was the first one to use dissipative particle dynamics to show electroporation. And what you see here is the membrane which is fluctuating on the left. And this is how simulations would show you biomembranes as they are fluctuating. And on the top figure, you see the green and the red are his water model, Drude model, which he developed. And you see that, and the middle white part is the bilayer membrane. I have not shown the lipid tails there. You see the formation of thread as initiation of electroporation. And in the lower video, you actually see this white pore that is the electroformed pore, which develops on application of electric field. So these simulations are under electric field. Okay. So we in my group look at giant vesicles as models for understanding cell electroporation, and we make giant vesicles by the electroformation method, and I think most of you know about it. So in this community, they are trying to develop giant vesicles as biomimetic objects for biological cells. Okay, so as you know, giant vesicles have this advantage that you have water inside, water outside. It's a nice spherical object. The lipids can be, you control the constitution of the bilayer. So unlike a cell membrane which has cholesterol, which has proteins and so on, you can put in whatever you want in the bilayer. But then the efforts are also to put inside whatever you want in a very controlled manner. So you might want to put genetic material, you might want to put actin, you might want to put cytoskeletal segments in a very controlled manner. And then what the object you get is as close as to a biological cell, and then it's much easier to study biophysics on a giant vesicle than in a biological cell. Those working in biological cells would know that they have a mood of their own, and they are not very conducive to study sincere biophysics. So in that pursuit, we try to look at giant vesicles as an object to understand electroporation. So if you look at the electroporation of biological cells, which is in the middle figure, you see that the cells are fairly well behaved after application of electric field. Now what I'll show is two cases of electroporation of giant vesicles, one on the left, one on the right. And they have some conductivity differences, they have some details which we need not worry at this point. But when you subject them to DC pulses, what you see is enormous deformation. In the left you saw this huge deformation. On the right it's even worse. You see that this vesicle undergoes a huge deformation, and like a Terminator, it actually remembers where it has gone. You will find that after some time it would recover back to its shape. Okay, so you would have thought that it's got completely destroyed. Now the question we asked in this work was, why do giant vesicles so extraordinarily large deformation? There is a huge amount of membrane disruption, membrane tubulation that happens in giant vesicles. This was studied in the literature as well, and several reasons were given, such as the poration in biological cells is better because there is cytoskeletal segments inside. The membrane is more rigid because of underlying spectrin in the red blood cells and so on. So let me just first go through one more round of observation. So which is so in this case, for example, when the vesicle deforms, you can plot the deformation as a function of time. And you essentially see three features here. So these are increasing electric fields. You keep on increasing the electric field and you see the deformation. And what you see is when the electric field is very low, you see so this is, you apply the electric field, the vesicle deforms, and then you switch off the electric field, it relaxes, okay, to a sphere. So this is the sphere. And when the fields are very low, they relax very quickly in order of around 2 milliseconds. When you apply stronger electric fields, they relax on the order of 10 milliseconds. And when you apply very, very strong electric fields, they would relax over order of 1 second or so. Now, this was discussed by Risk and Dimova, and what they suggested were 3 processes. So, when the fields are low, you don't have poration, and your vesicle relaxes over this time scale, which is the tension relaxes over membrane viscosity, and you get a time scale of around 100 microsecond to 1 second. So, this is unporated vesicle. If you have poration, then the pores close over a time scale of 10 milliseconds and you see relaxation over 10 milliseconds. And if your field is very strong, then in addition to the formation of pores, there is water efflux. The vesicle deforms and then it relaxes slowly over order of 10 seconds. So these are kind of rough calculations that you can present. And then what we can say is, if you are seeing very fast relaxation, you can say it is unporated. If it is relaxing over 10 milliseconds, you can say it is controlled poration. And if it is relaxing very, very slowly, then you can say that it is uncontrolled poration with a lot of tubulation. So we said that maybe the giant vesicles that showed excessive poration, unlike biological cells, what's the main difference between the studies that were conducted till then and the biological cells? And we realized that vesicles are typically formed in very low salt concentrations. Okay, and then one of the speakers also in the past mentioned that you would want to form giant vesicles at low salt concentration. Electroformation doesn't work at very high salt concentration. So we said, why not look at deformation of giant vesicles at very high salt concentration? And for that, electroformation doesn't work very well. So there is a method called gel-assisted method, where you actually form giant vesicles over a gel. We made it over a starch gel. So in the leftmost figure, you see a starch gel there on which you grow vesicles. Now you can have physiological salt concentration of the order of 150 millimolar or so. Okay. Now we then conducted studies with this system. So at low salt concentration, you see that the pores, the transmembrane potential slowly increases. This leads to formation of large pores. Okay. Whereas when the salt concentration is very high, your membrane charging time is very fast, your transmembrane potential increases immediately, shorts, and immediately falls. So we feel that in this, at high salt concentration, because of shorting of the electrode very fast, you probably have very controlled electroporation. So in the red thing that I have marked, on the left we have low salt concentration, on the right we have high salt concentration. And this is the aspect ratio, the deformation at the end of the pulse. And you see that with high salt, you see fairly well deformed vesicles. On the left with low salt, which was the status of the literature, you see larger deformation in the data below, but you also see a lot of tubulations, which are seen as those patches of membranes. We have confocal images, which I am not showing here, which indeed show that there is membrane disruption at low salt concentration. This is even better seen in the relaxation spectrum. So the top is low salt and you see the vesicles relaxing over order of 1000 milliseconds, which is 1 second. But when you go to high salt, you see faster relaxation, which means that the vesicles are having controlled electroporation. So we say that there is uncontrolled electroporation at low salt in the top versus controlled poration at high salt at the bottom. And another hypothesis which we have is that the edge tension, which Mahesh also was talking in the morning, he had a value I saw was of the order of 10 piconewton of edge tension. This value of edge tension increases if you have salt. So your pore closure is faster. So we say that at high salt concentration, you also have faster pore closure, which probably leads to controlled electroporation at high salt concentration. So I'll just quickly go through the next problem that I wanted to propose. Giant vesicles have been shown as a good model for understanding cell biophysics, and we argued that if you want to study the cell biophysics of nucleate cells, then they probably are not the best model. Something like this is not a best model, which we call as a simple giant unilamellar vesicle. We think that a compound giant unilamellar vesicle is a better model for eukaryotic cells. And so my student Rupesh, who is in the audience and who has a poster which was not very well received, I think, or not visited at least. So maybe you would want to visit in the tea break now. He made these giant vesicles by an improvised method. There's a method in the literature which he had to improvise quite a bit. So what it does is he forms a electroformed giant vesicle in a DMPC cholesterol system. It's extremely sensitive to composition and temperature. And once he forms the giant vesicle, he subjects it to osmotic shock so that it undergoes membrane remodeling. On the left, you see a stomatocyte kind of shape, and then there is further remodeling to form a compound vesicle. And he has this confocal image on the right, a DIC image at the bottom in the middle, which all confirm that it's a compound giant vesicle. So this is our model for a eukaryotic cell, where the inner vesicle represents a nucleus and the outer vesicle represents the outer cell membrane. Okay. So we did characterization of this using standard electrohydrodynamics. I'll not get into too much details of this. And the idea is that you subject these compound vesicles to different frequencies in AC electric fields. So you are applying very weak AC electric fields, non-porating AC electric fields, and you do a frequency scan. And what you see here is that the outer vesicle is deformed into a prolate shape, the inner vesicle deforms into an oblate shape, and you get this kind of a frequency map. And from this frequency map, we did this for a lot of study, a lot of cases, which is published since then. And what's important here is that for example, if you look at these transition frequencies, then from these transition frequencies, you can actually get the membrane capacitance through the expression that I've shown. So we suggest that this is one of the best ways to estimate both membrane mechanical properties as well as membrane capacitances of the nuclear membrane, because you cannot access the nuclear membrane by methods like micropipette aspiration, which Samir was talking in the morning. Okay. So having done this, we thought, can we electroporate now eukaryotic cells, in our case compound giant vesicles. So this is work in progress of Rupesh. So I'll tell you the physics, which I think is very interesting. You take a eukaryotic cell and you subject it to, just a minute, I'll just put the laser pointer. Yeah. So you take the eukaryotic cell, you subject it to electric field. Now the outer membrane, for example, if you subject to very low electric fields, weak electric fields, suppose 1 kV per centimeter, the outer membrane builds the transmembrane potential like this. It can go up to 1 volt, which means it can porate. But the inner vesicle goes only up to 0.2 volts, so it cannot porate, right? But if you apply, and so this is with 10 microsecond weak pulses, but if you apply, say 10 nanosecond pulse, very strong, with very strong field, for example, 100 kilovolts per centimeter, then you see that both the nucleus and the outer cell membrane can reach 1 volt of transmembrane potential. Yeah, so so both the membranes can get simultaneously electroporated, and this is what is used in technology called nanosecond pulse electric field. So in nanosecond pulse electric field, the idea is to porate the inner organelles. Example, in this case, this was the first demonstration in 2007, where they applied 100 kilovolts per centimeter of 60 nanosecond pulse electric field to Jurkat cells. And this is endoplasmic reticulum, which releases calcium, and the calcium sensitive fluorophore dye actually captures the calcium. So this was the first instance which tells that the inner organelles can be electroporated by nanosecond pulse electric field. Now we said that since the conductivity in biological cells is very high, the dynamics happens over nanoseconds. If the conductivity is very low, the same dynamics can be observed over microsecond or millisecond time scale. So what Rupesh did was conducted the same experiment in his compound giant vesicles with deionized water, very low conductivity, and he's done quite a bit of studies. I'm just presenting one sample result. So the idea here is, can we get the dye inside the nucleus? And his protocol, one of the suggested protocols of his, is two-step of electroporation. You put a very low electric field for 1 millisecond, 10 pulses, and you put the dye in the cytoplasm. And in the next step, he puts a very strong electric field, but a short one, which porates the nucleus and so the dye now can get inside the nucleus. And this is the intensity which clearly tells that when he puts the second pulse, the dye actually gets inside the nucleus. So I think I'm very sorry for overshooting the time, but these are the students who contributed to the work. Rupesh and Rakesh are here, and the picture of younger Samir is here, and he's been collaborating with me in some of the work. So, yeah, thank you.